Welcome back to the University of Melbourne Biology series. This is episode 13, and today we ask how the trillions of cells in a single body manage to act as one. Picture a sprinter at the starting line. A muscle in the leg has no idea what the eyes have just seen, and yet, in a fraction of a second, the whole body launches forward together. That only works because cells talk to one another, constantly, and because the body holds its internal conditions remarkably steady while everything around it changes. Over the next 12 minutes we will follow two great threads. First, how cells send messages, both as fast electrical impulses and as slower chemical signals. And second, how those messages are used to keep the body in balance, a property called homeostasis, in animals and even in plants. Let us begin with why communication matters at all. Start with the problem. In a complex multicellular organism, cell-to-cell communication is essential, because the cells must coordinate their functions and maintain homeostasis. No cell can act usefully on its own, it has to know what the rest of the body is doing. To solve this, organisms use two complementary kinds of signal. The first is chemical, where one cell releases a molecule, such as a hormone, that another cell receives. The second is electrical, generated by neurons, where the signal travels as an action potential. These are not rivals, they are partners. Many of the most important responses in your body hand off from one to the other, electrical to chemical and back again. So our plan is simple: understand each system on its own, then see how they combine. We will start with the electrical signal, because it is the faster of the two. Electrical signals are the work of neurons, and the signal itself is called the action potential. It is an electrical signal that a neuron generates to transmit information. How is it made? It begins with a change in the membrane potential, driven by the influx of sodium ions into the neuron. As sodium rushes in, the inside of the membrane swings sharply positive. This phase is called depolarization. It does not last. Almost immediately, potassium ions flow out of the neuron, and that efflux brings the membrane back down again, a phase called repolarization. The membrane then briefly overshoots, dipping below where it started, in a phase called hyperpolarization, before settling back to rest. So the whole event is a tightly ordered sequence: sodium in to go up, potassium out to come down, then a short undershoot. That single spike is the basic unit of every electrical message in the nervous system. Generating one spike is only half the story. For the message to be useful, it has to travel the length of the neuron, and this is propagation. The action potential is propagated along the axon, the long projection that carries the signal away from the cell. Now here is where biology adds a clever piece of engineering. Many axons are wrapped in an insulating sheath called myelin, and that sheath is interrupted at regular intervals by small gaps known as the nodes of Ranvier. Myelination and the nodes of Ranvier together enhance the speed of propagation. Rather than crawling smoothly along every point of the membrane, the signal effectively leaps from node to node, and that makes it dramatically faster. This is why a signal can race from your spinal cord to your foot almost instantly. Speed, here, is the entire point. The electrical system exists to be fast. Now to the second system, chemical signaling. Here a cell sends its message as a molecule, and the receiving cell must have the right machinery to detect it. Chemical signals come in several types, sorted by how far the message travels and which cells it reaches. An autocrine signal acts back on the very cell that made it. A juxtacrine signal acts only on cells in direct contact with the sender. A paracrine signal reaches nearby cells in the local neighborhood. And a hormonal signal travels long distance to act on far-off target cells. A hormone is the classic example of that last category. Notice the pattern: these four types are really one idea seen at four different ranges, from a cell whispering to itself, out to a cell broadcasting across the whole body. Chemical signaling is how cells reach targets that an electrical wire could never directly touch. We now have both systems on the table, so let us compare them directly, because each has a job the other cannot do as well. Electrical signals, the action potentials carried by neurons, are rapid, they propagate along neurons and deliver a message almost at once. Chemical signals, such as hormones, are suited to long-distance communication, and they can be paracrine, autocrine, or hormonal in their reach. Both are essential, and both serve the same ultimate purpose: coordinating cellular activities and maintaining homeostasis. Think of it as the difference between a phone call and a letter posted to everyone in a city. The electrical call is instant but point-to-point, the chemical letter is slower but can reach a vast audience at once. A well-run body uses both, choosing speed when speed matters and reach when reach matters. And that purpose, maintaining balance, is exactly where we turn next. All of that signaling exists for a reason, and the reason is homeostasis. Homeostasis is the maintenance of a stable internal environment in a multicellular organism. Your cells can only function within narrow limits, so the body works constantly to hold conditions steady even as the outside world swings. The way it does this is through feedback mechanisms, and these come in two forms: positive and negative. Negative feedback loops are the workhorses of homeostasis. They counteract a deviation from a set point and pull the system back toward it. Positive feedback loops do the opposite, they amplify a process rather than damp it down. Communication sits at the heart of all of this, because feedback only works if the parts of the body can signal to one another using exactly the chemical and electrical mechanisms we just met. Let us look at each kind of loop in turn. Start with negative feedback, the loop that keeps things stable. A negative feedback loop is a regulatory mechanism that counteracts a change in a system by producing a response that tends to reverse that change. If a value drifts up, the loop pushes it down; if it drifts down, the loop pushes it up. To do this, the loop uses three components working together: a receptor that detects the change, a control center that processes it, and an effector that carries out the corrective response. The classic example is the regulation of blood glucose. There, two hormones, insulin and glucagon, work as a negative feedback pair to keep glucose levels in balance. Body temperature regulation works on the same principle. The defining feature to remember is simple: the response always opposes the original change. That opposition is precisely what holds the internal environment steady. Not every loop is about holding steady. A positive feedback loop is a regulatory mechanism that amplifies a process, often leading to a rapid and sometimes excessive response. Instead of opposing a change, it reinforces it, driving the system further in the same direction. Because of that, positive feedback does not maintain stability, rather, it drives a process to completion. This makes it less common in homeostasis than negative feedback, but it is critical in certain physiological events. Two clear examples are blood clotting and childbirth. In each, once the process begins, the response feeds back to intensify itself until the job is finished, the clot is formed, the baby is delivered. So the contrast is sharp. Negative feedback is the brake that keeps a value near its set point, while positive feedback is the accelerator that pushes an event all the way through to its end. Communication and homeostasis are not unique to animals. Plants do it too, and they have evolved their own complex mechanisms for cell-to-cell communication, which are essential for maintaining water homeostasis. The showcase example is the plant's response to water stress. When water is scarce, the plant relies on a hormone called abscisic acid. Abscisic acid plays a key role in responding to drought by closing the stomata, the tiny pores in the leaf, and so reducing transpiration, the loss of water vapor. How does it work at the cellular level? The signal is carried through a signal transduction pathway, and the response involves the efflux of ions and then water out of the guard cells, the pair of cells that flank each pore. As those guard cells lose water, they go slack and the pore closes. It is a beautiful parallel, a chemical signal, driving a response, all to hold an internal variable steady. Step back and look at the whole picture. Both of our threads turn out to be one story. Cells communicate using two complementary modes, the rapid electrical signal and the longer-range chemical signal, and both of these are essential for coordinating cellular activities and maintaining homeostasis. Homeostasis, in turn, is the maintenance of a stable internal environment, achieved through feedback, and feedback is nothing more than communication arranged into a loop. Negative feedback uses that loop to counteract deviations and hold a set point, positive feedback uses it to amplify a process to completion. And the same logic reaches across kingdoms. A plant closing its stomata with abscisic acid is running the very same idea, a signal producing a response that keeps an internal variable in balance. From a sodium ion crossing a membrane to a stoma sealing shut in a drought, it is all communication in the service of balance.